Hello and welcome back to Paleocast. My name is Dave Marshall and this is episode 101 on the organic preservation of Dinosaur Bone with Dr Evan Saitta of the Field Museum in Chicago. If this is your first time listening, Paleocast is the complete paleontological podcast, in which we present the whole paleontological field and discuss organisms and time periods not covered in the regular media. We don't read and recite, but we invite scientists onto the show to tell the story of their own research in their own words. In this instance, we're talking about molecular taphonomy, which sounds pretty intimidating, but you should be able to follow along if you remember your high school chemistry. If not, we've got a lot of supporting images on our website, which provide a great visualization of the results, so make sure you visit paleocast.com to check those out. For those of you who've been following the show over the years, you'll know that we've long coveted one of the People's Choice Podcast Awards, and voting is now open for 2019. In the entire month of July, you can nominate Paleocast in the Science and Medicine category at podcastawards.com. If we get enough public nominations, we make it through to the final 10, with the winner chosen by a panel of podcasters, journalists, and listeners. In order to nominate a podcast, simply go to podcastawards.com and we'll put links in our social media channels, and there you can register with an email address, nominate your favourite shows in each category, and that's it. And we really do appreciate any nominations we receive. It really means a lot to us. Finally, please like and share this episode on social media in order to help us reach as many people as possible. And comments, reviews, and requests are always welcomed. Hi Evan, thanks so much for coming on the show today.
Hello, thanks for having me, Dave.
Okay, against my better judgment, I think we should introduce you to our audience. So, can you start off by telling us how you got into paleontology?
Well, it's not really a super interesting story because I've been interested in paleontology my entire sentient life, basically. I don't remember a time when I wasn't interested in this science, and my parents did a good job of encouraging that interest. They had a rule that if any book that I wanted to buy as a kid, they would always get it for me, and that helped fuel my passion for science. I found my first fossils out on the beach in Florida when I was maybe 6 or 7 years old. One of my first dinosaur dig in high school. Studied biology and geology in my undergraduate degree at Princeton, then went over to the UK at the University of Bristol for my master's and PhD in paleobiology.
And that's where we met.
And that is where we met and became good friends.
And what would you be doing if you weren't a paleontologist?
Also, probably another really boring answer because it would be some other type of science. It'd be biology of some sort.
Really?
Yeah. I really do like the natural sciences. I mean, maybe, to branch out a little bit farther, I guess engineering is kind of interesting, having a little bit more hands-on thing. But yeah, I love the interdisciplinary aspect of paleontology in my work. So I'm very interested in biology and geology and chemistry. So there's plenty of other fields of science other than paleontology that I find fascinating.
And you wouldn't have made it as an athlete?
No. I did track and field for a little bit up into on my college career, but no, I was never intending on becoming an athlete.
Too slow, like the rest of us.
Exactly. Yes.
So you eventually ended up working on taphonomy and dinosaurs. We know what dinosaurs are, but what is taphonomy? Can you explain what it is and why it's important?
So taphonomy is the study of all of the physical, biological, and chemical processes that happen from the time of an organism's death all the way to its discovery as a fossil. So it's a very complex, multi-factored process. And the reason that it's important is because we think paleontology is important. So, as paleontologists, we study the history of life on Earth, we study the history of evolution, and our data set for that analysis is the fossil record. And so, if you want to make or attempt to make conclusions about the history of life on Earth, you need to properly interpret the fossil data. And of course, if you don't know how the fossils form, then that's going to influence your ability to study them. So you need to take into account the sort of preservational biases that go into becoming a fossil, so that you know what is preserved, what isn't preserved, just so you can read the fossil record accurately, really.
So which other wider disciplines does taphonomy draw upon? Has it got a bit of chemistry, physics, maths, everything in there?
Well, it's definitely chemistry heavy. It's also quite biology heavy. So, there's the biochemistry of the original starting molecules. There's a lot of microbiology if you're talking about how things rot and decay. There's a lot of organic chemistry and geochemistry, right? So the modern emphasis of taphonomy is studying organic molecules specifically. And which organic molecules can preserve in fossils and for how long they can preserve in the rock record. And so very often you'll have people who aren't necessarily taphonomists, but will collaborate with taphonomists who are organic geochemists. So think along the lines of oil and gas formation, that sort of branch of science. And I mean, there's a reason why we call it fossil fuel, right? These are organic fossils, oil and natural gases. And so the way that many taphonomists, particularly myself, like to view taphonomy and fossil biomolecules is in the same way that an oil and gas scientist might study the formation of oil, right? And so, so there's a large chemistry component in it too. But you can also focus on different aspects. And so some researchers, especially historically, have focused more on the sort of sedimentology aspect. So this is, finding a dinosaur quarry, for example, and looking at the layout of the bones and whether they were quickly buried or exposed to the surface, whether they were transported by the action of water, so on and so forth. And so, strong chemistry side, strong geology side, and sedimentology side. But it depends on what part or what stage of the process you're looking at, whether you're looking at early taphonomic influences or more late taphonomic influences.
Sure. Well, regardless of which stage we're looking at, taphonomy, I see it as like, I guess the fossil record as a whole is like trying to read a book that has vast numbers of pages torn out and whatnot. And so can it tell us anything new, or can it only ever be a quantification of what's actually missing?
Well, it's both, I suppose. I mean, if you have a new understanding of how fossilization works, and this is something that I want to begin to get into, is you can then begin to predict what sort of information you can get out of the fossil record. And so that's kind of, I think, the cusp of where we're at right now is we're starting to understand what types of biomolecules are capable of becoming fossilized and which ones break down more rapidly. And then we can attempt, with that understanding, to go and look for those signatures in the fossils. And so there's all sorts of new insights that can provide us. So a good example is the work of Jakob Vinther, who we both know very well. He was my PhD advisor. And he was the one who realized that melanin, the pigment melanin, can fossilize. And so now we're beginning to reconstruct the color of dinosaurs. And this was something that people had suggested was impossible for quite a good number of years. And so, an understanding of the chemistry of fossilization, not only can it help you avoid misinterpreting the fossils, suggesting that, something is preserved which really isn't, but it can also guide you towards new evidence, and that can give you new insight onto life in the past.
Brilliant. Well, speaking of new insights, we're going to be talking about dinosaur bones today. So why did you choose to study the taphonomy of dinosaur bones?
So the taphonomy of dinosaur bones in particular was an offshoot of some of my other work. So my PhD focused on how soft tissues preserve in these so-called exceptional fossils, these exceptionally preserved fossils like the feathered dinosaurs you have in China, where the feathers are preserved as these sorts of flat, thin, carbonaceous stains around the bones. And when I was examining feathers, I was looking at not only genuine fossils, but I was also doing some experimental work derived again from oil and gas scientists. And in that work, you use high heat and high pressure to simulate what's called diagenesis. So this is the high heat, geothermal heat and high pressure from the overlying sediments that a fossil will experience when it's buried deep underground for millions of years, right? And so you're trying to speed up that process. It's kind of like pressure cooking, really, is the analogy. And so what had happened is when I was looking at these structures, which rather than collagen protein, which we'll get to in a moment, it's keratin protein. So this is the protein that makes up hair and feathers and scales. And we began to establish this model where the keratin protein components of something like a feather became unstable at high heat and high pressure and broke down and were ultimately lost from the fossil, whereas the more stable components, such as melanin pigment, as well as some calcifying salts that you can get in some of these structures, those remained behind. And it appeared to be a pretty good model of fossilization. So, last summer I published a paper where we basically can create synthetic fossils that even down to the level of a scanning electron microscope resemble some of these like feathered dinosaurs from China with these carbonaceous stains that are composed of this melanin pigment. And so we had this, what we thought was a pretty well-supported model for how things like feathers fossilize, and it involved the breakdown of proteins and the loss of the proteinaceous material. But this is in quite a bit of contrast to quite a lot of high-profile reports of dinosaur collagen. So like keratin protein, collagen protein is relatively stable, at least in comparison to other types of proteins. Collagen is predominant in tissues such as your skin, but it's also in cartilage and in bone. And so there had been claims using different techniques, particularly, and we can get into this in a little bit, some mass spectrometry techniques, as well as what's called an antibody staining technique, claiming that you had not only the presence of dinosaur collagen in fossil bones, but that they were essentially pretty pristinely preserved in order to elicit the responses they were getting, the sort of signatures they were getting in their analyses. And this is in contrast to the model of feather fossilization that we had. And there's also the question of the mineral component of the tissue. So unlike the feather keratin, which is largely exposed and unprotected, the collagen in the bone is within a mineral component, right? It's within the calcium phosphate, also known as apatite, of the bone. And whether or not this provides any sort of shielding is people are skeptical of it, but it was still fairly up for question, I suppose.
Well, I think we've covered so many different points of your research there. I'd like to take a step back, if we could, and just explore what kinds of things we would actually expect to find in a dinosaur bone when the dinosaur was alive versus when we find it in normal conditions, and then finally onto what people have suggested that they've actually found in there.
Well, so in life, the dinosaur bone is highly mineralized, so calcium phosphate, large calcium phosphate component. In terms of the protein component, the dominant protein is going to be collagen. And of course, the ratios of protein to calcium phosphate mineral vary depending on the bone, so it can influence like the sort of biomechanical or physical properties of the bone. So something like deer antler is going to be different than other types of bones. And then within the bone, you'll also have some structures such as blood vessels. You'll have some bone cells called osteocytes. And that's largely what you would expect to find. Obviously, the blood vessels would have originally had blood cells in them. But that's largely the sorts of organic components within the bone.
And it's not too dissimilar to what other animals have in their bone, is it?
Oh, yeah. So if you're asking dinosaurs specifically, yes, it would be very similar to pretty much all vertebrates, and including humans. Right. So this is, the bone tissue is going to be relatively conserved in that sense, if you're just talking about, calcium phosphate and collagen protein. So it's going to be similar to like a modern chicken bone, modern human bone, that sort of thing. But now here's the thing, though, in terms of the rest of your question is what you would expect to find. Well, the sort of perspective that an organic chemist or a geochemist might take is that proteins and some of the cell membranes of these osteocytes, these bone cells, are unstable, particularly under high heat. So the bane of biological molecules, one is temperature. So think back to high school chemistry. You increase the temperature, a lot of chemical reactions will then increase the rate at which they occur, but also water. So a lot of these complex organic molecules are what's called polymers, where they're individual constituents stitched together in a chain. So for example, DNA has nucleotides stitched together into a sequence. Protein has amino acids stitched together into a sequence. And the way you can break up the sequence and basically cleave it is something called hydrolysis. So you basically insert a water molecule in between the sequences, and then the sequence degrades and fragments. And so there's also a few other types of degradation reactions that occur in these biomolecules and in proteins. So, for example, when the amino acids are freed from their chain, they can flip their configuration. So certain molecules, because of their structure, they have what's called chirality. And so they can either occur in one or two mirror image forms. So they have like a left-handed version and a right-handed version. And life, the vast majority of life, tries to keep all its molecules on the same page. So they try to keep all of their molecules in the left-handed configuration. But over time, things like amino acids, once they are no longer being produced by these organisms, they will equilibrate within a closed system into a 50-50 mixture of this left and right-handed form of the molecule. And that's another type of degradation that occurs in proteins. And this is not to confuse people, but it's important in terms of the analysis that we did in this study. So it's probably good to cover that. And so you have all these reactions that break down things like cell membranes or proteins. And so the expectation, or I should say like maybe the conservative position, would be that you would not expect tens of millions or hundreds of millions of year old dinosaur protein in cells. So starting in the 90s and really kicking off in the mid 2000s, there were claims of dinosaur proteins, dinosaur blood vessels, dinosaur cells. And of course, these attracted a lot of attention, both from the scientific community as well as the popular media. This was feeding right off of the back of Jurassic Park, essentially. And so a lot of interest as a result. And those studies, basically what they did was demineralize the bone. So, for example, if you put the bone in an acid solution, the mineral will break down and it will reveal whatever organics are inside. So whether it's the collagen protein that's helping to form the structure of the bone, or any blood vessels or cells inside. And then these exposed organics were treated with antibodies. So antibodies are immune system proteins that can bind to certain targets. So, for example, they can bind to a specific region of a target protein based on its three-dimensional structure. So it's sort of like a lock and key. They fit together. And so antibodies, which were designed to detect collagen, were adhering to these structures. There was some mass spectrometry data, which appeared to have some very short sequences consistent with collagen. And almost immediately, these results were met with debate by other researchers. So, for example, the sequence data from mass spectrometry was challenged as being statistically insignificant or potentially a result of laboratory contamination. And then there's also some question as to how specific these antibodies really are and whether they might be binding to molecules other than the collagen target. And so last fall, I published a paper where we reanalyzed one of these dinosaur feathers, which antibodies had suggested consisted of keratin protein. And we found basically no organic signature. It was inorganic calcium phosphate producing the fossil feather. And also it was covered in a cyanoacrylate superglue, right? And so it's a compound, it's a glue that's used normally in preparing fossils in the traditional sense. And it's also used in medical research to adhere to antibodies and transport them to a particular target. And so,
Wow.
Yeah, and so we were then skeptical that these antibodies were yielding true positives. We were thinking that these are actually false positive results, and there's actually kind of a litany of other molecules that might produce a false positive with these antibodies, some of which would be expected potentially in ancient organics, right? So things that are more akin to what oil and gas scientists might be dealing with than what a biochemist studying a living organism is dealing with. And as a result of all this, we, in terms of my keratin, my feather fossilization research, in terms of some of the debate around these bones, I figured it would be good to address the question myself and use some of the techniques that I was using for studying feather fossilization. And that's where it leads me to this project.
Awesome. Well, we'll get into details of that in a second, but I'm really interested in what some of the reports that have come out have included, because some of them get really massive claims. Like, I'm sure that there's been red blood cells preserved, blood vessels, other things like that.
Yeah, there have been some big claims. The blood cells have been one. There's kind of the blood cells are kind of their own sort of, taxonomy there in terms of the diversity of these structures. So there are some structures which appear like these little red dots within some of these demineralized vessel-looking things, like these blood vessels. Others have identified the vessels as potentially coming from a biofilm of microbes. So it's sort of the microbial community plus any of their secretions that form these structures. And so the blood vessels and some of the blood cells even have been suggested to actually be microbial biofilms living within the porous cavities of the bone. The supposed red blood cells within those vessels have alternatively been identified as either pyrite or iron oxide framboids. And so these are minerals that you'll very often find in ancient sediments or even within fossil bone itself. There was another couple of claims, I guess 2 claims of red blood cells in fossil bone where the structures were actually organic. And I was able to, using some of these high heat, high pressure experiments, to essentially recreate those structures and their full ranges of shapes and sizes, essentially. But rather than dinosaur blood, it was really just degraded turkey skin that we used. And so what looked like these little blood cells were probably just degraded organics folding under the vacuum of a scanning electron microscope. So when you look at these things under scanning electron microscope, you have to pump down to a vacuum, and we suspect that that is actually leading to the deformation that looks like blood cells, right? So you were seeing this sort of continuous sheet of organics that was folding into these various structures. And so you have the potential not only for microbial contamination to produce something that looks like a dinosaur soft tissue, but also a completely abiotic process to produce something that looks like a dinosaur soft tissue, like a red blood cell. And so, yeah, there's a lot of different possibilities, and it's not just because the scientists are trying to ruin our dreams of Jurassic Park. It's because the original claim is very unexpected. It is not consistent with our understanding of the rates at which these chemical degradation reactions occur, right? It's not consistent with what we know about how proteins degrade. And now, I would be changing my tune maybe if somebody found a T-Rex frozen in ice, but the youngest T-Rex is still over twice as old as the oldest polar ice sheets, so I don't think that's going to happen anytime soon. But yeah, so there's plenty of alternate explanations for a pretty extreme claim. So to put it in perspective, the oldest, I would say, non-controversial or uncontested protein sequences or peptide sequences are not even 4 million years old. The oldest collagen, relatively intact collagen sequences are from 3.4 million years old, but they're from bones that are preserved in permafrost up in the high latitude Arctic. So they've been refrigerated. There's 3.8 million year old protein sequence, which is this very, very short protein sequence, so only part of the protein has survived. And it survives in this very unusual mechanism where it adheres to the mineral of the eggshell. So it's found in bird eggs, and it adheres to the mineral component of the eggshell, which then stabilizes it, and it actually has this sort of strange molecular cooling mechanism, which I won't necessarily get into. But point being is you either have to keep things frozen or you have to have some very, very unique preservational mechanism. And even that has only so far gotten us to 3.8 million years old, which is 1 or approaching 2 orders of magnitude younger than these supposed dinosaur collagen. So it's an enormous gap.
Yeah, 3.8 to, what would it be, 65 million?
Well, so I believe some of the oldest, at least among the recent literature, the recent studies, some of the oldest claims are Jurassic collagen, where they used what's called spectroscopy in order to detect it. And that would, I believe those specimens are like maybe 220 million years old or something like that. I can't remember. But yeah, so yes, but the youngest non-bird dinosaurs are, or no, it'd be probably 250 million years old. Anyways, youngest non-bird dinosaurs are 66 million years old. So yes, quite older than the non-controversial protein reports.
Okay, and have there been any other studies that have looked at any other animals other than dinosaurs? Because it sounds too good to be true that you go back 3.8 million years, you still got these proteins, nothing for what's basically 60 million years. And then all of a sudden you get all of these dinosaur organics being found in all over the place. So what happened in between, and has anyone looked at any other groups?
Well, so that's really more a gap of the research that has been done. I mean, the reason why there's the gap is people, like to study dinosaurs, and so the reports were focusing on dinosaurs, and people just haven't really done a more systematic approach. And now there are some researchers that are, especially the researchers doing the much younger proteins that are working from the present and then going back in time. And that's how we're getting to 3.4, 3.8. Another approach done by some others is to basically jump into the deep end of the pool, as has been said, and go to dinosaurs, which you could argue whether that is an effective approach or not. But it's really just in terms of what has been reported. I don't think anyone's claiming that there's a gap per se, even if they do believe the dinosaur collagen reports.
Okay, and other groups, has it always just been vertebrates?
The recent literature has focused on vertebrates. It's not always dinosaurs. Quite a variety of different species have been examined. But I mean, this is coming from myself, who also tends to focus on dinosaurs. I mean, there is the temptation to go to dinosaurs just because it's a rather exciting area of paleontology. So, you do a project on a dinosaur bone, you can put a press release out for it. If you do the same study on a fossil shrimp, you probably can't. And it's an unfortunate bias, but it probably is true. And so, yeah, a lot of attention has been done to dinosaurs.
And that's why Paleocast exists, to readdress the balance. So, Anyway, let's talk about dinosaurs now. You recently have done some work looking at the dinosaur bone, and to test a lot of these claims that have been made. So, how did you go about your work? What kinds of hypotheses were you testing, and how did you do it?
Yeah, so the question is essentially, well, there's kind of two parts. It's like, do you detect signatures of ancient organics, particularly proteins? Do you detect any evidence consistent with the organics being what's called endogenous to the bone itself? Or do you see evidence for exogenous material infiltrating the bone? And so that's kind of where we're getting at. Do they look ancient, and do they look like they're deriving from the bone? And so my approach was to collect fossils from scratch, essentially. And so I went out to Dinosaur Provincial Park in Alberta, Canada, to a site where there's literally more bone than rock at the layer producing bone. And basically, you spend the morning removing the overburden and then the afternoon collecting the samples. And the way I collected the samples were to use aseptic equipment. So it was a Centrosaurus bone bed. This is a horned dinosaur about 75 million years old, related to Triceratops from the Cretaceous. And after removing the overburden, so the overlying rock from a region of the quarry, I would begin to use more and more aseptic equipment. Once you find a bone, you expose just enough of one end to realize what the bone is and where it's going. You can then take out, rock and all, the rest of that bone using sterile equipment, sterile foil. You can then seal it off into a sample bag and put it into a cooler to keep it refrigerated. You then try to keep it as cool as possible under refrigeration until it's brought into the lab, and we brought them to various labs. And we ran essentially a battery of different analyses on the bone. Anything from structural analysis of what happens when you demineralize the bone and what sort of structures are residing within the bone mineral, to basic aspects of what sort of elements, chemical elements, are making up the bone, all the way to more complex studies of the chemistry, and then leading into finally more biological analyses where we attempt to characterize any microbial community that's living within the bone. So we kind of go over a range of characterization, I guess you could say, of what's inside the bone.
Well, results in a second, but I would put to you that having dug up the bone, you've already contaminated it.
Yes, exactly. So there's a good reason why we don't claim that these are sterile collection techniques. These are aseptic collection techniques. And so anytime you're taking what's called environmental samples, so this is a very common thing now with what's called environmental genomics or metagenomics, there's an understanding that the very process of collecting the sample is going to alter it. There's the main goal, however, is to not pretend that you're in some sort of sterile environment, but to rather reduce the contamination that you're introducing, or that you could introduce to the bone. And now the problem with paleontology is that the field techniques have changed practically not at all for the last 100 years or so. And so, no, I do not claim to make any sort of statement that these bones were collected in a sterile manner. However, the results from the various analyses we got were consistent. They were fairly easily interpretable. And they were suggestive that, yes, the aseptic protocols that I used were sufficient. The idea that there was literally no alteration to the samples is obviously not true, but it appears to be sufficient for our purposes, yes.
Was it difficult doing the field work in a hazmat suit?
Well, no. So it wasn't really a hazmat suit. It was more just making sure that I didn't sweat on the bones, making sure that I was wearing clean gloves, that the tools were clean, that I wasn't breathing or talking or spitting onto the bones. It was, field work is always very hot and arduous, so there's that, but I'm used to that now. But yeah, so it seems that I was successful in not profusely sweating on the bones or anything like that. So we seem to have gotten usable data.
Okay, so let's talk about your results. What did they suggest?
So the results show that one, and we have sort of a negative and a positive aspect to this. So the negative aspect is we don't see any evidence for ancient proteins. For example, the structures that are removed from the dinosaur bone when you demineralize it do not resemble either younger subfossils from like thousands of years ago that have collagen preserved, or modern bone collagen. The elemental analysis is also not consistent with collagen. Various chemical techniques, so for example, something we use called pyrolysis, gas chromatography, mass spectrometry, which is a bit complex and I won't go into it. Those chemical fingerprints don't match with collagen protein. And most importantly is the amino acid analysis. So this is going back to some of the preface that I established earlier. So what you can do is you can actually, and this is something that's recommended to do before you attempt to sequence anything in order to determine whether you have anything worth sequencing. You can take the sample and you can study the composition or the profile of the amino acids within it. And so if you have a sample that is supposedly full of dinosaur collagen, you should have an amino acid profile that matches that of collagen. We do not. Furthermore, if you have very ancient proteins, or even if you just have very ancient amino acids disassociated from the protein, as in other words, if the protein has fragmented, you would expect what's called racemization to occur. And this is going back to the left-handed, right-handed forms of the amino acid. And so if you have a very old sample of amino acids, you would expect fairly high amounts of right-handed configurations of these amino acids. We don't see that. We see very high levels of left-handed configurations. And this is consistent with recent contamination. So taken together, you don't have a collagen profile and the amino acids are recent. Very strong evidence that you don't have dinosaur collagen. We also did a few other techniques. So we used radiocarbon analysis, and it's probably important for the listeners to know that, this is not an attempt to radiocarbon date the bone, right? Because in a few minutes, we'll talk about how the end result is that we think the bone is an open system, so you can't really do it. You can't do radiocarbon dating, but you can analyze the radiocarbon in there. And so what we found is that there is non-radiocarbon dead organic carbon. In other words, recent carbon in the bone. That's also a sign that you don't have a ton of dinosaur protein and that something else is infiltrating it. So the amino acids, the radiocarbon, and then one of the more interesting things that we found, and also the total amount of carbon is higher in the bone than the surrounding rock matrix, right? One of these really interesting things we found, though, was that the amount of DNA inside of the bone is like 50 times higher than the amount of DNA in the surrounding rock matrix. And that right there is very strong indication that there is some sort of microbial community living within the bone. Even though,
but why? We've got carbon in the bone where we expect to find carbon. We got DNA in the bone where we expect to find DNA. Why isn't that coming from the dinosaur?
Well, and it gets to this idea of open versus closed system. So if bone was a closed system, then this would be very peculiar results, because we would be finding high levels of DNA in a sample that's, 75 times older than it should be. But the sum total of the evidence that we have is more consistent with microbial infiltration of the bone. And this is not really surprising, especially, and I know you have a lot of listeners who do paleontology themselves. When you're out looking for fossils, especially dinosaur fossils, like out in the Badlands, it's very common to find like lichen growing on bones. It's very common to find plant roots growing through dinosaur bones. These fossil bones are sources of phosphate mineral that can attract life. So life needs certain minerals, in particular phosphates. So phosphates are used, for example, in your ATP molecules, which are your kind of like metabolic currency within your cells. They're used in your DNA, in the sugar phosphate backbone of your DNA. And so life needs phosphorus. And so it's not surprising to see bone, which is also chock full of vessels and porosities and medullary cavity, become infiltrated by life underground that's seeking these nutrients. The bones, again, are also porous, which not only allows microbes the ability to enter and to live inside of them, but it can also wick up moisture. So, for example, I'm sure some of the listeners are aware of the sort of trick to identify fossil bone. If you put it to your tongue and it sticks to your tongue through capillary action, it's likely to be fossil bone instead of rock. That's a good example of the bone's ability to wick moisture, and of course life needs moisture. And so what we're thinking is that these microbes are seeking refuge essentially inside of the fossil bone. And so you see all this recent organic material. It's not consistent with dinosaur material, but it's consistent with modern microbes. And so if you actually look at the microbial community using what's called 16S rRNA sequencing, so you're looking at you're using sort of the RNA, so which is like DNA, to barcode the different species of microbes that are living inside of a particular environmental sample. If you look at that, not only are the microbes preferentially living in the bone compared to the surrounding rock, but it's also a different community of microbes living in the bone. And so there's something very unusual going on. For example, 30% of the microbes living inside of the fossil bone that we looked at while it was underground are related to something called Eusebia, this bacteria which, as far as I'm concerned, or as far as I know, has only been reported on the skin of sea cucumbers and in Etruscan tombs. And so it's this very poorly understood microbe, but it's making up 30% of the community in the bone. So it's exciting for the microbiologists because we have this new habitat that's probably present all over the earth, deep underground, with some very unusual microbes, and we don't know yet exactly what they're doing in these bones, what sort of metabolisms they have, what sort of diversity do we have between a bone, say, found in Alberta, Canada, and a bone found somewhere else in the world. And so it's very interesting to the microbiologists for sure, but it's very worrying to the paleontologists because it means that if you're going to try to detect anything originally from the dinosaur in these bones, it could, even if it has a signature of being ancient organic material, it could be bacteria from the Cretaceous, it could be bacteria from 10 million years ago, it could be bacteria from 500 years ago. And so the open system of bone not only means that microbes can enter and complicate our attempts to identify anything original in the dinosaur, but anything that breaks down in the dinosaur can then be lost, right? It can leach away from the bone.
So do these results suggest that all the previous discoveries of dinosaur organics were misidentification of contaminants, or is there still room for any of these to, that haven't been entirely ruled out?
Okay, so a couple things. One, the word contaminant used is very interesting because normally, and especially in terms of this debate about dinosaur proteins, contaminants are very often used in the sense of laboratory contaminants, right? So somebody studies an ostrich or an alligator collagen sequence and then studies a dinosaur, and maybe there's cross-contamination just within the lab. This form of contamination, though, is a bit more pernicious, I suppose, because it is contamination within the ground, right? And so it goes right at the heart of this idea that dinosaur bones are supposedly some sort of pristine time capsule, right? And so even if so we were talking about, the inability to collect dinosaur bones in a perfectly sterile way. Well, even if you could come up with the technology to do that, it would be kind of a moot point because the bones have been interacting with subsurface microbes for who knows how long, maybe even tens of millions of years. And so the point you make about contamination is very interesting. As for, we got to kind of go through the semantics here. As for whether this means there's no hope, some hope, or lots of hope, we got to be careful about what molecules we're talking about. Different biomolecules have different fossilization potential. So, for example, we were talking about fossil melanin. That appears to be very thermally stable. Steroids also appear to be very thermally stable, and we've detected fossil steroids from hundreds of millions of years ago. Oil and gas are also technically fossil organics. They're clearly very stable deep under the ground. If your question is dinosaur protein, then I take a hard line. I don't think we have any reason to suspect that dinosaur protein, in the sense of amino acid sequences being preserved, will preserve in bones of that age. There's some workaround. So there's been suggestions, and I agree with the underlying chemistry. There's suggestions that you can have a naturally occurring version of what happens during cooking. So when you cook meat or bake bread, you have these browning reactions, and that is proteins break down, sugars and fats break down, and then those breakdown products condense together to form these sort of thermally resistant brown sticky polymers, right? And we know that those form actually in the geologic record and that they can persist in the rock record. But again, if you're talking about bone, well, you're talking about an open system. So even if you have ancient browning molecules from within side of a dinosaur, how do you know that's not Cretaceous bacteria or bacteria from 10 million years old? You know, the open system of bone also complicates this. I don't want to say that there is nothing original even within open system dinosaur bones. And that's because some biomolecules are not only more thermally stable, or at least can be converted into a thermally stable form, but some are less soluble, which means that they might not have the propensity to leach away from the bone. And so, for example, in our study, we found what's called kerogen, which is basically when fatty acids, such as the fatty acids potentially in cell membranes, become separated from the rest of their molecule, which is the polar head of the molecule. They can then link up together to form these hydrocarbons, and this is more or less what we mean by kerogen. And we detected that in the bones. And there's a possibility that that is genuine dinosaur hydrocarbon. It could also be kerogen deriving from ancient bacteria, which is the problem. But I wouldn't necessarily rule out that there is nothing dinosaur in the ancient organics within bones. The question of dinosaur protein, however, I'm going to take a hard line and say no. But there's the interesting question, though, then becomes some of these structures, right? So the vessels and the cells. I see a few ways of preserving those that are actually dinosaur derived, but they're not unaltered in the way as has been suggested. One is the lipids form this sort of kerogen, so these hydrocarbons, and those basically then form in situ, in place, and so they preserve what looks like vessels or cells. Another one is these browning molecules essentially doing the same thing. But again, the problem goes back to the open system. It could be biofilms. It could be microbes that then infiltrate what are or what were completely void spaces in the bones, and then they form these secretions and colonies of bacteria that then make a mold of the vessel. So, for example, one of the vessels that we looked at in the dinosaur bone had infilling, and there was appeared to be a greater carbon content in the interior of the vessel than the exterior. This is not consistent with a blood vessel being somehow stabilized or undergoing fixation. It's much more consistent with microbes coming and growing within inside of the cavity. And again, even some of these degraded molecules could be ancient bacteria rather than ancient dinosaur. And so, yes, it's not encouraging for the taphonomy side, for the paleo side.
Well, you've only looked in really in depth in that one place in Alberta, which I don't think is a concentrate Lagerstätte, is it? It's not a site of really special preservation, more so concentration of bones. It's a bone bed, as you were saying, it has more bone than it does rock. What about some of the extraordinary fossils that we found, say like, was it a dinosaur tail that was preserved in amber? Is there any chance of finding organics in that kind of environment that we know has a lot? It would be a lot harder to permeate. It'd be a lot harder to have that natural, use the word contamination again.
So a few things. One is a lot of these reports of dinosaur collagen are coming from places like Alberta or Montana. And so we were comparing similar environments. Two, we do plan on expanding this study in the future and looking at the chemistry of bones from other localities. With regard to your mentioning of amber, amber is interesting, and there's actually relatively little work done on preservation of fossils within amber. A lot of that has to do with the fact that if you ask a museum curator if you can destructively analyze your dinosaur tail in amber, they'll tell you to take a hike. I have some experimental work, so I was talking about my high heat, high pressure experiments where it appears, we haven't quite worked it out entirely, but it appears that we can produce something that is like a synthetic copal or maybe even an amber, and that we plan to in the future to use an experimental approach to study amber fossilization, because if we put feathers in amber, we can destroy as many of them as we want because we created them. So there's some future work to be done there. There's actually a recent paper which did this sort of amino acid analysis on feathers in amber. And I'm not super convinced by the results. It was sort of mixed, and even in their paper, they kind of admit that it's sort of mixed results. At the very least, it's not consistent with like protein preservation or high sequence preservation. At best, it's probably consistent with some of the original amino acids being trapped within the amber. And so there's a couple problems with that. One, it takes a long time for amber to fully form. So resin goes to copal and then goes to amber, and it takes a long time to actually have the full polymerized amber. You have specimens from, I think it's like 10 million year old or maybe even 25 million year old fossil resins that are not fully polymerized as amber. They're technically subfossil copal. And so it takes a long time for presumably the system to close. Amber is an organic matrix itself, right? So there could be contamination from the ancient plant even. It tends to attract all sorts of things. So if you look at amber specimens, sometimes you can have, several different fossils preserved within the same piece of amber. My suspicion, though, is that the breakdown of molecules in amber occurs as we would expect, but that amber probably is a relatively closed system. And so I expect to see thermally stable molecules, the things that we would expect to find in fossils, but that they might be more likely to be trapped if the amber forms rapidly enough, I suppose. But I guess the short answer is we really need more work on that, on amber.
Given the really high-profile media reports, each time there was a purported discovery of the blood cells and vessels and whatnot, what's been the media response to your negative results?
Well, I mean, we only published the paper last week, and I've still been talking to various reporters and podcasters and whatnot, and so it's hard to say.
We're not the first podcast.
No, unfortunately not. Go and listen to, what's it called? Skeptic's Guide to the Universe? I don't know.
Yeah, great. Everyone go do that.
Yeah. Hey, you need a little competition. It'll force you to do your best.
I don't think we're much competition for them.
Yeah. I actually had never listened to them before, so
Had you listened to us, though?
Oh, of course. Yeah,
Of
I was a
course.
true paleocaster. So it's hard to say. I can say from experience that it is an uphill battle. I think that science journalists need to take a little bit more responsibility in terms of attempting to cover rebuttals to papers. So they'll publish on an exciting new find, but if there's a subsequent paper later on and they recognize, hey, I covered the original study, last year, it would be nice for them to cover the discussion. And it's not just because of sour grapes or anything. It's an understanding of what science is. Science is competing hypotheses, right? It is a constant process getting hopefully closer and closer to truth. It's not just a series of eureka moments. And in fact, I'm actually kind of surprised in the I guess we're maybe beyond the age of reality television, but people love a good, argument in a fight. And, sometimes some of these scientific debates are actually very interesting. It makes you think about the process of evidence and analysis and logic. And it's the process that every scientist goes through, even if they're not engaged in a sort of debate against a different researcher, right? So if you are the lone scientist working in your lab, within your own head, you have various alternate hypotheses for the explanation of what you observe, and then you wrestle with those and you eliminate some of them, and hopefully you come down to one. It's the same process, even if the different alternate hypotheses are in two different heads, right? And so it is about the process of science. And so it would be nice to have coverage of science in action rather than just the process of science. And, that's kind of, I think, the main hurdle of science education or science outreach is a lot of people view science as, here's a science textbook, this is what it says. But it's not, it's a process. And the debates in science are where that process is alive.
In your work with accelerating the fossilization process that you spoke about before, you had some surprising reactions to that within the media. Can you tell us about those, and specifically some of the creationists that took it on board?
Yeah, so what's interesting is that a lot of this work tends to draw some very peculiar crowds. So, for example, this dinosaur protein work, creationists, or specifically young Earth creationists, love the idea that dinosaur proteins are being preserved in these bones, because to them, it's not representative of some new exceptional chemical preservation mechanism. To them, it's an indication that the bones are actually much younger than what the evolutionists claim that they are. And so so there's this strange latching on to dinosaur proteins. So they probably don't like my most recent paper, in the sense of I am not in support of ancient dinosaur proteins, but they did like and I sort of colloquially call it synthetic fossilization. So we're in a laboratory trying to use essentially a high powerful pressure cooker to simulate deep burial over shorter periods of time. And they like that work because, it's the sort of idea that, if the scientists can do it in 24 hours, then what could a omnipotent being do in 6,000 years? Clearly, the omnipotent being would not need 75 million years, as the scientists claim. And so they liked that. I had gotten a few requests to talk about it with them, and I try to be polite, and I usually give a short little spiel about, the sort of evidence that we have that the Earth is not 6,000 years old, and that the dinosaur bones are, 70 or more, a million years old and whatever. But there's only so much you can do when someone is kind of possessed by an ideology. And so the best thing is to just be polite. And I try not to give too much attention to them.
Do you still see your research being discussed in those fields?
I have recently. Well, I got an email from somebody not too long ago, but yeah, I think they'll cover it. They got their giant ark out in Kentucky or wherever. I'm sure they're having a great time out on the ark with some of my work, but yeah, you can't think about that. I mean, it's just people are going to misrepresent you, and that's how it is.
It's a shame. But looking ahead to the future now, what is next for you? What's next for the taphonomy of dinosaurs, and what's next for taphonomy in general?
Yeah, so I think the question of taphonomy in dinosaurs and taphonomy in general is the same question. We gotta, kind of come to that understanding that as exciting dinosaurs are, we have to treat them the same way that we treat any other Mesozoic fossil, and we have to think of them. If we're studying their chemistry, we have to think of them as chemists, not as dinosaur scientists. If you ask the microbiologists what they're excited about, and we're actually planning on doing work on this in the future, is examining more of these dino microbiomes, as I've been calling it for lack of a better word, and seeing what they're doing in the bone, how they're living in the bone, how they're getting there, whether or not they might explain some of these soft tissue reports, what sort of diversity of dino microbiomes do we have? If we dug up a fossil elsewhere in the world, would it show a similar community or a different community? So they're very excited. In terms of paleontology and taphonomy, though, I got asked this a little while ago. They said, so are you convinced about dinosaur proteins? I mean, I'm not going to spend much, if any, time trying to investigate dinosaur proteins more. I do not believe personally that you can get protein sequences from Mesozoic fossils. I have a project that'll hopefully be published sooner rather than later, which further solidifies that, but I can't really talk about it as of yet because it's not peer-reviewed or published. But and so proteins I'm not too thrilled about. However, we're entering a different age of paleontology. So for the history of our science, we've been going out to different areas of the world, and we've been looking for bones and teeth and shells to hopefully maybe find a new species or something along those lines, or to find an exceptionally preserved skeleton, like an articulated skeleton. But we're beginning to enter a very different type of fossil hunting. We're looking for molecular fossils now. It's not just teeth and bones. We're looking for fossil molecules. And we know that there are some classes, some categories of molecules that can survive deep into the fossil record. And they could tell us all sorts of things about the biology of these extinct organisms. And so what I want to begin doing is to, in a combination of experimental work and also direct observation of fossils, try to predict and then hopefully find various fossil biomolecules that might allow us to understand the biology of organisms in a new light, right? And to study it in extinct animals in ways that we never could before. So the good example is fossil melanin, right? So we now know that melanin can preserve deep into the fossil record. I think the oldest record is like 300 million years old or something along those lines. And it allows us to reconstruct color patterns and hues of extinct animals. This is something that we thought was completely impossible. And we're now beginning to get insights into things like camouflage or sexual display. And it's an exciting new, it opens up a whole new body of evidence, essentially. And my interest, my goal, is to try to find as many of these molecular fossils as we can. And I think that's probably the future of taphonomy.
That sounds brilliant. Thanks so much for joining us today, Evan.
Yeah, good to talk to you again.
Paleocast was brought to you by Dave Marshall, with Joe Keating, Laura Soul, Liz Martin-Silverstone and Caitlin Colery. It was made possible by funding from the Paleontological Society and the Paleontological Association, but the show now relies on contributions from you, the listeners. If you've liked this episode, please consider donating, and thanks to everyone that's helped out so far. We'd also like to thank the Ocean Collective for use of their music. Please visit paleocast.com for the supplementary material to this episode and for our archive of past programmes. Subscribe to our Twitter feed @Paleocast and like us at facebook.com/Paleocast to get all the latest news.